Hello everyone. In this video, we are going to see the screening of recombinant clones by colony PCR. The colonies after transformation are patched on an agar plate, and this plate will be further used to carry out the colony PCR. So this picture shows a plate with transformed colonies and a patched plate. So the plate with patched colonies is open in the laminar airflow to pick the colonies, as you need this plate for further confirmations. Like if you got positive in colony PCR, so you can again take the colony and go for further confirmation like plasmid isolation, restriction digestion, and you can also give for sequencing. So to do the colony PCR, first you need to arrange sterile appendiform tubes and pick the colonies from the plate by using sterile toothpick or sterile micropipette tip. So according to the number of the colony, you have to label the appendiform tube with the same colony number. So you can add, after picking the colonies, you have to add 50 microliter of sterile distilled water to the tubes, and then you have to mix the colonies. After adding the colonies to the tubes, you can mix as shown in the video. Then you need to vertex the tubes, and then this suspension is used for colony PCR. So this suspension used is as a template for the colony PCR. So to set up a colony PCR reaction, PCR master mix is prepared with Taq polymerase, water, buffer, DNTPs and primers. You can make a PCR master mix and then you can aliquot into the PCR tubes. So on the screen the components are given: sterile distilled water 12.8 microliter, buffer 2 microliters, DNTP 1 microliter. So this is for a total reaction of 20 microliter, and from the suspension we are using 5 microliter of the template. First you need to aliquot the master mix into the tubes, then you can add the template to each tube. So template will be 5 microliters of each colony. So there are two ways to do this colony PCR. One is by mixing the colony in sterile distilled water and vortexing and using it as a template. The other method is you make the PCR mix a total volume of 20 microliter, pick up the colony and mix it in the PCR reaction mixture. So directly you will pick the colony and add it into the mixture. If you are following this method of using sterile distilled water, you will be not getting much debris, like you will use 5 microliter of template. If you are directly picking the colony, you will have lot of debris and all. So after setting up the PCR reaction, now the tubes were placed in the thermal cycler and the reactions were set. You can use blue colony for negative control. Thank you.